Hi, everyone. Happy Friday. Thank you for joining us today. Today is Friday, July 22nd, and we are here to meet the DM drug developer, Harmony Biosciences. I'm Tanya Stevenson, the CEO of the Myotonic Dystrophy Foundation, and we are delighted that you are joining us. A couple of things about the Myotonic Dystrophy Foundation. Our vision is new this year, and we want to make sure everyone is very clear. We envision a world with treatments and a cure for myotonic dystrophy. The mission of MDF is community, care, and a cure. We support and connect the myotonic dystrophy community. We provide resources and advocate for care. We accelerate research towards treatments and a cure. A reminder of some of the resources and support available from MDF. Toolkits and publications available at myotonic.org/toolkits-publications. Our support programs, including support groups throughout the US, Canada, and in different languages, myotonic.org/find-support. Our calendar of events, myotonic.org/calendar/month. And our digital academy, where we host all of the videos and presentations of our past events, including today's presentation, which will be shared probably in the next week or so. So you can try myotonic.org/digital-academy for today's presentation later this week. Today we are hearing from Harmony Biosciences. This is part of our Meet the DM Drug Developer sessions. It's a series that's been happening the first Friday of the month at 12 o'clock Pacific. We have one more this year before our conference in San Diego, and that will be August 5th, and we will be hearing from PepGen. You can register and submit questions in advance at myotonic.org/meet-DM-drug-developers. We also have one more Ask the Expert session. So the third Friday of each month this year at 12 o'clock Pacific, we have been hearing from experts in different areas and specialties. On August 19th, we will hear about disability rights, future planning, and special needs. Register and submit questions at myotonic.org/ask-expert-series. Okay, you are here to meet Harmony. One of those team members is Rachel Radomski. Rachel is the head of Patient Advocacy. She leads Harmony's work with people living with rare diseases, their caregivers, and patient advocacy organizations across the rare disease landscape, including MDF. The patient advocacy team at Harmony works to represent the voice of people living with a rare disease across Harmony and develops innovative solutions and programs to meet the dynamic needs of the communities that Harmony serves. Rachel and her team are honored to live Harmony's core value of keeping patients at the heart of everything they do at Harmony Biosciences. Also joining us today is Dr. Bill Jacobson. Dr. Jacobson is the Senior Director of Clinical Development at Harmony Biosciences. He's an experienced drug developer and clinical trialist, originally trained as a neuroscientist with almost 30 years of experience in the pharmaceutical industry. Dr. Jacobson works on strategic and tactical aspects of drug development, and his current focus is on rare diseases of the central nervous system. He's a clinical lead on Harmony Biosciences' development program for type 1 myotonic dystrophy. If today's program speaks to you, please consider showing an appreciation with a donation to MDF. You can find our donate page at myotonic.org/donate. Okay, it is my great pleasure to welcome the Harmony Biosciences team. Thank you so much.
Hi, Tanya. All right, let me share my screen here. All right. Hi, everyone. Tanya, thank you so much for that kind introduction. It's an honor and a privilege to be here with the Myotonic Dystrophy Foundation today. It's been wonderful to get to know and work with Tanya, you and your team, the board, and the whole myotonic dystrophy community over the last several years at Harmony Biosciences. We're thrilled to be with you here today. We're going to take you through a brief introduction to Harmony Biosciences. We did do one of these last year. It was our first Meet the Developer with MDF. But if you haven't gotten a chance to watch that or get to know a little bit about Harmony, I'll provide a brief overview. And then I'm going to introduce my colleague, Bill Jacobson, our head of clinical development for myotonic dystrophy. And he's going to provide an update for our clinical trial. And I don't want to steal his thunder, but I know we're excited to share some changes we've made to the trial with the community today. And then, as Tanya mentioned, we will do our best to take as many questions as possible over the next hour. So again, thrilled to be here today and really looking forward and excited to seeing hopefully many of you at the conference this fall. I know we haven't gathered together as a group, I think, since 2019. So we're looking forward to attending that meeting in San Diego, as well as share more about Harmony with you all today. So very briefly, if this is the first time you've heard of Harmony Biosciences, this is who we are. So we're a pharmaceutical company headquartered right outside of Philadelphia, Pennsylvania. And what we're working on, innovative therapies for people living with rare neurological disorders who have unmet medical needs, including myotonic dystrophy. So we're a relatively new company. We were established in 2017 by Paragon Biosciences, and we licensed the rights to pitolisant, which is the investigational medication that we're exploring in myotonic dystrophy from our French partner, Bioprojet. So we became a public company in August of 2020, and we have about 200 employees now. So if you look to the bottom right of the slide, this is our clinical development pipeline at Harmony. So you'll see myotonic dystrophy on there. Bill will share a little bit more information about our phase 2 clinical trial in myotonic dystrophy today. And this is the team that I get the pleasure to work with at Harmony. Myself, my colleague Gabrielle. So at Harmony, we specialize in developing and delivering treatments for rare neurological diseases that others often overlook. And so one of the things that we really strongly believe is that where empathy and innovation meet, a better life can begin for people living with neurological diseases. And our team across Harmony comes from a wide variety of disciplines and experiences. And our work is driven by our shared conviction that innovative science translates into therapeutic possibilities for patients who are at the heart of everything we do. And so the work that we do on the advocacy team, we collaborate with national rare disease organizations, patient advocacy groups like MDF. We also provide support via our Patients at the Heart, Progress at the Heart, and patient advocacy organization funding opportunities. And as Tanya mentioned when she was reading my brief bio, we also work to incorporate the patient and caregiver voice into all levels of our work at Harmony. And I'll talk a little bit more about how we did that in the myotonic dystrophy community last year, leading up to the initiation of our phase 2 trial. And if you have any questions or would like to reach our team, we're always available at advocacy@harmonybiosciences.com. And so one of the things that Tanya was sharing is how strongly I and our team at Harmony believes in listening to people living with rare diseases and their caregivers. And so when we were looking into past efforts to people living with myotonic dystrophy to find shared symptoms in their lives, we really looked into across the landscape that you see below, the Christopher Project, which is a survey many of you may be familiar with, the FDA, the MDF-led patient-focused drug development meeting that took place in 2016, several sessions at previous Myotonic Dystrophy Foundation conferences that are focused on CNS, and then a patient and caregiver advisory board that we held last year as we got ready to launch our phase 2 clinical trial. And so all of these important reports and meetings pointed to a very common theme, that many people living with myotonic dystrophy reported that they experience excessive daytime sleepiness, fatigue, and or cognitive dysfunction as a part of living with myotonic dystrophy. And so I mentioned briefly we hosted last year a patient and caregiver advisory board for 6 advisors who shared their daily lives with us, their struggles, their fears of living with myotonic dystrophy, and members of our clinical team, our medical team, and our advocacy teams were engaged over a period of 2 days. And during that time, many advisors shared with us that while other symptoms of their disease had a significant impact and they felt were important, many felt that they weren't as disruptive to their daily lives as sleepiness, fatigue, and cognitive dysfunction. So overall, the advisors that we engaged shared what a significant burden that these secondary symptoms are on the quality of their lives, and that they felt that they weren't well managed with available treatment options. And so this is just a snapshot into key insights that emerged when we were looking at patient-reported experiences. So as you can see from the quotes that are below, there are several themes that come through here that have a profound impact on people living with myotonic dystrophy and their caregivers. So Christopher Project, there's a quote here that says the experiences of patient respondents varies across different types of the disease, but people living with DM1 reported more daytime sleepiness, more impact from fatigue, and a harder time staying alert. And so then this quote is from the FDA patient-focused drug development meeting. More than anything, I want to recapture the joy of life that can be lost to a neuromuscular disease that requires most of my energy to get through the day, leaving little reserve for the things that really matter. The next quote comes from a Myotonic Dystrophy Foundation conference in 2017. I hope, beg, and pray for a drug or therapy that will help all the brain-related symptoms that DM1 patients are experiencing. And then the final quote comes from the advisory board that I mentioned we held last year. If I can go to a movie in a wheelchair, that's okay for me, as long as I'm able to stay awake for the movie. And so our team was really moved by hearing these different experiences. And we feel that there's, a strong desire in this community for a new potential treatment option that could potentially alleviate some of the symptoms associated with EDS, fatigue, and cognitive dysfunction. And so with that, I'm happy to turn this over to Bill Jacobson, my colleague, who's going to explain a little bit more about what Harmony is exploring in patients living with type 1 myotonic dystrophy.
Thank you, Rachel. And I'd like to second Rachel's welcome to all of you. As she mentioned, it's an honor and a privilege to be able to talk with you a little bit about what we're doing to try and address some very important and meaningful aspects of myotonic dystrophy. In addition to being an honor and a privilege, it's a pleasure and a privilege to be able to do this. As Rachel mentioned, at Harmony, patients are at the heart of everything we do, and having the opportunity to speak directly to you is something we all take great pleasure in. So if we could move forward, Rachel, to the next slide. I'll talk to you just a little bit about some highlighted aspects of the disorder itself, which I'm sure many of you are quite familiar with, but this will kind of set the stage for what I'm going to talk to you about in terms of how we are addressing this. So myotonic dystrophy is a multisystemic disease, and while certainly people who do not know anyone who has the disorder or do not have the disorder themselves often focus on the muscular aspects because of the nomenclature of myotonic dystrophy. It is truly multisystemic, and it affects many, many parts of the body, not only the muscles, but the heart, the gut, the eyes, the endocrine, and several other systems. But very importantly, from our perspective, myotonic dystrophy is in part a brain disorder. And again, while people tend to focus very often on the non-muscular symptoms, or I'm sorry, on the muscular symptoms, very often the non-muscular symptoms actually precede the muscular symptoms, and they occur in early onset adult disease. And there are a large number of non-muscular things that can occur. These include such things as early cataracts, which are quite unusual, but things like excessive daytime sleepiness, fatigue, and some form of cognitive dysfunction, amongst many other things. So we're focused to a large extent, not exclusively, but certainly from a major perspective on sleep. Sleep, as you all know, is a naturally, periodically recurring state of inactivity. It's characterized by the lack of loss of consciousness, reduced responsiveness to external stimuli. We are actually focused in our studies on wakefulness, which is the absence of sleep and is marked by consciousness, awareness, and activity. Sleep and wakefulness are very intimately related, but they are in fact controlled by different systems in the brain. One of the things that we are focused on because of the nature of the drug that we're investigating, and I'll talk about this in a moment, is a brain chemical or a neurotransmitter referred to as histamine. And this is the same drug or biochemical that's involved in allergy and so on when it's impacting the peripheral or non-central parts of the body. But we're really looking at and studying what histamine does within the brain. And amongst other things, it's intimately involved in the control of sleep and wakefulness. We're looking at an investigational medication called pitolisant. And pitolisant has been shown to clearly impact the amount of histamine within the brain. And as a consequence of that, there are many other chemicals in the brain that are involved in wakefulness, attention, and cognitive function that are impacted as well, with histamine being an intermediary. So there's a lot of interest here in how, by manipulating the amount of histamine in the brain, we can impact some of these non-muscular symptoms that, as I'll show you shortly, have significant impact on the lives of people with myotonic dystrophy. We can go to the next one, please, Rachel. So this is a very interesting piece of a study that was done by Dr. Chad Heatwole and his group, largely at the University of Rochester. This was a study called the PRISM-1 study. And what it did was it looked at the prevalence and the impact of various symptoms in type 1 myotonic dystrophy. They've also done a subsequent study called the PRISM-2 study looking at type 2 myotonic dystrophy, but our focus is on the type 1 myotonic dystrophy. So there are a number of things that are non-muscular symptoms in myotonic dystrophy that are highly prevalent. In other words, large numbers of patients with the disorder suffer from these problems. And you can see that in the blue bar graph that's shown here on the left. In addition to looking at the prevalence or how frequent and common some of these symptoms are, we also looked from the perspective of the patient at how impactful some of these factors are. And you can see that certainly problems with the arms and hands are very, very common. You can see that fatigue and impaired sleepiness and some difficulty thinking, which you see a little further down, are actually quite prevalent. As a matter of fact, fatigue and sleepiness are amongst the most prevalent symptoms in type 1 myotonic dystrophy and frequently appear before muscular symptoms do occur. Importantly, though, is if you look at the impact factor, in other words, how much does the appearance of one of these symptoms impact the lives of an individual suffering from the disorder, you can see that fatigue and problems with sleepiness are quite high. As a matter of fact, fatigue actually has greater impact than do the muscular symptoms with arms and hands. And we find the same for excessive daytime sleepiness. So the takeaway from here is that while it is certainly important, and there's a tremendous amount of research that is ongoing looking at the causality of the disorder and the muscular consequences of the disorder, the myotonia, the limitations in mobility, the muscular pain that can occur, and so on. The non-muscular symptoms really do have tremendous impact, and those are the symptoms that we are focusing on primarily in our clinical work. Next slide, please, Rachel. So when we do a study, we have to have an idea of why we're doing it. What is the objective of the study? And in our case, in the particular study that is ongoing, we're interested in evaluating the safety as well as the efficacy of pitolisant, comparing it to placebo, to an inactive drug, a so-called sugar pill, in treating the excessive daytime sleepiness in adult patients with type 1 myotonic dystrophy. Now, again, that is our primary objective. That's the first and, in a sense, most important objective that we're looking at. In order to actually study an objective, we have to have a methodology. There's something that we have to measure, and these are called endpoints. So in this case, with the primary objective that I have just outlined to you, our primary endpoint is the change in a certain score called the Daytime Sleepiness Scale, which we refer to as the DSS, from the beginning of the study until the end of the study. And we'll be comparing how that changes when we compare pitolisant to an inactive medication, to a placebo. Next slide, please. So as I mentioned, there are, actually, I'll get to that point in a moment. I do want to just explain a little bit about the Daytime Sleepiness Scale because it's a relatively new endpoint and measurement. The DSS is a so-called patient-reported outcomes. In other words, it's the patient who are telling us what it is that we want to learn. It's a questionnaire. It's a fairly short questionnaire, 5 questions, where patients are asked to evaluate their degree of sleepiness. And the higher the score, the greater the degree of self-assessed sleepiness. So what we are hoping to see is that when we compare our active drug to an inactive placebo, that the score decreases to a greater extent. The DSS was developed in adult patients with myotonic dystrophy by Dr. Luc Laberge and his group up in Saguenay, Quebec, Canada. And this is a very interesting area because due to what is called a founder population, where there was a fair amount of inbreeding within a group of a population that had the genetic signals for the disorder, the prevalence of type 1 myotonic dystrophy in the Saguenay region of Quebec is approximately 10-fold higher than it is in the general population. So it's a very, very important research center. They have a very large cohort of patients there that are treated, and there's a fair amount of research that has ongoing there. The instrument has been studied extensively, and it has been validated from a number of different aspects to ensure that it is actually measuring what it is that we think we're measuring and that we need to measure. And since it was developed in the French language, it is actually being linguistically validated, and that process is essentially completed. So we are going to be certain that we can assess the perspective from the patient as to whether or not pitolisant had any beneficial impact on their sleepiness in a manner that will be recognized and accepted by the regulatory authorities, who ultimately have to assess both the safety and efficacy of pitolisant in our target population, adults with type 1 myotonic dystrophy. Next slide, please, Rachel. So as I did mention, and I was beginning to before the last slide, in addition to our primary objective, we have a number of secondary objectives that we're evaluating within the study, looking at other important symptoms of myotonic dystrophy in addition to sleepiness or sleep propensity. So we're studying fatigue, which is different from sleepiness. We're using an instrument called the Fatigue Severity Scale, which was developed in actually originally for multiple sclerosis, but has been validated in a number of other disorders. We're looking at cognition, elements of what is sometimes referred to informally as brain fog, and there are many, many aspects of this that we could look at, but we have made a decision to look at those specific domains of cognition that seem to be primarily and largely impacted in patients with muscular dystrophy. So these include things such as psychomotor function, or the speed of processing, how quickly you can think. And we're using various computerized cognitive assessment tests for each of these things. We're measuring attention using another computerized scale, and we're measuring working memory using still a third scale. Very importantly, these next two assessments that we're using are extremely important. The first one is called the CGIS, or the Clinical Global Impression of Severity Scale. And this is a scale where the clinician himself or herself, actually looks and tries to assess through a number of observations how much excessive daytime sleepiness is actually present. At the same point, we're very interested in, and the regulators are very interested in the patient's impression of how sleepy they are. So we're going to be measuring that using a similar but distinct scale called the Patient Global Impression of Severity Scale. And both of these scales are going to be tied to the excessive daytime sleepiness symptomatology. Finally, we're looking at the overall burden of disease. And this takes in 17 different areas or themes that patients with myotonic dystrophy can have problems with. Certainly, not all patients have problems with all of these areas. In addition to looking at things like sleepiness, fatigue, and cognition, they also look at muscular function, gastrointestinal symptoms, and so on and so forth. The instrument is actually very heavily weighed towards assessing sleepiness and fatigue and cognitive function. So this will give us an idea of the overall benefit or lack of benefit, should that be the case, of our drug on the overall burden of disease. The Myotonic Dystrophy Health Index, commonly referred to as the MD-HI, was also developed by the research group at the University of Rochester with Chad Heatwell and his group. Next slide, please. So we also have an exploratory endpoint. You know, most of the endpoints that I've just described to you are subjective, reported. There's no actual hard measurement. However, we're very interested in doing some objective testing. So we're actually using a test called the Maintenance of Wakefulness Test, and looking at, pardon me, change from the beginning of the study until the end of the study for patients who consent to this. This is an optional test where we'll be comparing pitolisant to placebo. This is a type of sleep study. It's performed in a sleep laboratory, and it measures how long one can actually stay awake under certain well-defined conditions. So in other words, after a night of undisturbed sleep, we put the patient into a quiet room with no stimulation, and we just observe them and see if they can stay awake for up to 40 minutes. And the sleep latency is the amount of time that it might take for them to fall asleep. We do this test 4 times, 4 sessions over the course of the day, and we take the average. And this is an objective measurement that has been used, for example, to measure wakefulness in long-distance truck drivers or in airline pilots, where it's very important to stay awake. So we are also using this as an objective measure, but it is exploratory and it is optional, so that patients who are unable to, for instance, take the time, because this requires at least a full day, will still be able to enter the study and participate if they meet the other entry criteria or requirements. But we are very interested in collecting this data, and you know, are urging our sites and potential patients to take the test if they are able to. Next slide, please, Rachel. So the study itself, this is a phase 2 study. It is a relatively small study with a total of 78 patients that we are targeting to recruit. They will be randomly assigned in a blinded manner to one of 3 treatment groups. A third of them, or 26, will be on a higher dose of pitolisant. Another third will be on a lower dose of pitolisant. And the last third will be on inactive placebos. And again, this is a double-blind study, so the investigators will not know what the drug assignment is. The patients will not know, and we will not know. Nobody will know unless it becomes necessary to unblind a patient for some reason. We will not know the distribution of patients who is on what drug until the study is over and we have locked our data, are unable to change it. We will then unblind and do the appropriate analyses. Next slide, please. So this is an overall schematic of the study. We start off by doing a screening period where we bring potential patients in, patients who have an interest and seem to be eligible, and we screen them. We do certain tests to ensure that they meet the eligibility criteria for the study, and we'll talk about those in a little bit. Those that pass screening will then move on to a titration phase, which I'll discuss in a moment. I would like to tell you that the screening period can last up to 6 weeks. It certainly does not have to last up to that long, but the reason we have allowed for such a long screening period is some of the assessments that need to be done can take a bit of time. We need to do a genetic test, and that can take a couple of weeks to get the results. We need to collect data on sleep in a sleep diary for at least a week or 10 days, actually, up to 10 days, and so on. So screening can actually take place in approximately 2 weeks, but it can also last up to 6 weeks. If an individual passes the screening part of the study, they will then be entered into the study where they will undergo a blinded 3-week titration where the dose of the drug will slowly be increased up to the maximum that they might be taking. They will then be randomly assigned to, as I've mentioned previously, either a low dose or a high dose of pitolisant or placebo. At that point, they will enter into an 8-week dosing period where the dose will not be changed. Now, this entire period is double blind. In other words, nobody will know what drug anyone has been assigned to. At the end of the 8-week stable dosing period, the double blind period, patients who remain eligible will be offered the opportunity to participate in an open label extension phase. In other words, this will be a phase where everybody has access to the drug itself. There won't be any placebo. We will, since we did not know who was assigned to what particular dosing during the double blind period, we will retitrate everybody to make sure that they undergo a proper titration with the drug. And then they'll all be entered into the open label phase where the criteria for continuation and participation are much less stringent than they are. We do this for 2 reasons. One reason is certainly to provide everyone who has participated in the study, whether they've been on active drug or not, to actually have access to the drug. So if you've perceived benefit, you may want to continue. And if you haven't perceived benefit, that may be because you were on placebo. It may also be because you just didn't respond to the drug. We won't know that. But the opportunity is here, should you choose to enter into the open label part. So that's one reason we do this. The other reason is this allows us the opportunity to continue to collect safety data with long-term use of the drug. Obviously, people will be exposed to the drug in the double-blind phase for no more than 11 weeks, 3 weeks on titration and 8 weeks on stable dosing. Now, I would like to stress that we do have a fair amount of safety information on this drug, but in different patient populations. This drug has never been administered, at least in a clinical trial, to patients with myotonic dystrophy, and they are a very unique patient population. So we are very interested in collecting longer-term safety data, in addition to the original efficacy data that will ultimately form the basis for our submission to the regulators for approval in this patient population. Next slide, please. So we'll talk a little bit about eligibility criteria. In other words, who can enroll into the study. Rachel, next slide, please. So we have what are called inclusion criteria, things that you have to meet, and exclusion criteria, things that would exclude you from participation. So we're looking at adult patients with myotonic dystrophy, so individuals between 18 to 65 years of age. We are doing a genetic test on all of our potential patients to look and ensure that they all have the appropriate genetic signal indicating that they do have type 1 myotonic dystrophy. Many of the patients who are interested in participating, who or are participating have actually had these tests done, but we are doing them in everyone to ensure that we use the same methods and the same sensitivity to ensure that we are not enrolling patients who do not have the disorder. As I've mentioned, we're interested in sleepiness, so these patients have to have moderate or severe excessive daytime sleepiness, and that is something that is assessed by the study doctor. We are, because of the consequences or the potential consequences on sleep and sleepiness and fatigue, we are not allowing patients who are currently using either CBD or cannabis products for at least a month prior to the study, and they have to agree to remain off of these drugs, so they either have to have not been using them or to wash out for about a month. We are allowing patients who are currently using a wake-promoting agent. There's nothing that is approved for the treatment of excessive daytime sleepiness in type 1 myotonic dystrophy. There are drugs, however, that are used in other disorders where sleepiness is a problem. And some practitioners actually prescribe these to their patients, and some patients actually see good benefit from these. Those patients obviously would not be suffering from continued excessive daytime sleepiness. But if someone is taking one of these wake-promoting agents and is still suffering from excessive daytime sleepiness, we are allowing them to participate in the study as long as they've been on a stable dose for a while, and as long as they continue to maintain that stable dose throughout at least the double-blind portion of the study. We're looking for patients who are able to walk independently with or without an assistive device. So if a patient uses a cane or a walker or a brace, these are all allowed. What we're trying to avoid in this initial study, because this is our first study in this patient population, are patients who are wheelchair bound, whose disease has progressed to that point. And hopefully we will be able to look at them in the future should this study be successful. As is often the case when we're looking at investigational medications, appropriate contraceptive precautions for women of childbearing potential are required. And then obviously, in the opinion of the investigator, of the treating physician, the patient is capable of understanding what is required and following the protocol on the administration of the oral study drug. Next slide, please. Now, you can actually go to clinicaltrials.gov and look up this study and see a full list of all of our inclusion and exclusion criteria. We do have a large number of exclusion criteria. I'm not going to go through all of them. I'll just touch on some of the key ones. There are a number of things that can contribute to excessive daytime sleepiness, and one of them is sleep deprivation or inadequate sleep. So we're asking all of our potential patients during the screening period to do a sleep diary, where we will look at the average amount of sleep that they're getting per night, and obviously want to ensure that their sleepiness is not being contributed to primarily by a lack of sleep. There are also certain sleep-related breathing disorders, such as obstructive sleep apnea, that are common in this group, and they can contribute to excessive daytime sleepiness. We are certainly not excluding anybody with a sleep-related breathing disorder, as long as, in the opinion of the investigator, that sleep-related breathing disorder is not a main contributory factor. So in some cases, we will know this by medical history or previous sleep testing. In some cases, some of our investigators are actually assessing sleep with a sleep test to see if obstructive sleep apnea or other similar disorders are actually contributing to the sleepiness. We don't want patients who have participated in a study with another investigational medication for at least a month before. We have to make sure that they've washed out of any medication that might be confounding. In addition, we are excluding any medications that might interfere with the action of pitolisant. So there are certain drugs that interfere with the way the body metabolizes or handles pitolisant, and those are excluded and so on. Since this is a population with excessive daytime sleepiness, they tend not to suffer from insomnia, but on occasion they do. So we are allowing occasional use, not more than twice a week, of any sleep-promoting treatments that could affect excessive daytime sleep. So things like sleeping pills, like Ambien and so on, are allowed with limited use, but anyone who is using these regularly or nightly is not allowed to partake in the study. And then finally, similar to the last criterion in the inclusion criteria, if there is someone that the investigator thinks for any reason, unstable, other systemic unstable disease or inability to follow the directions and so on, those patients need to be excluded. And lastly, there are a number of standard exclusion criteria that we see in many clinical trials that have to do with your kidney function, your liver function, women who are nursing, breastfeeding, or planning to during the course of the study are excluded. Excessive caffeine intakes, and we have this well-defined, certainly, the use of moderate amounts of caffeine, regular coffee intake is not exclusionary. The use of any prohibited medication, and anyone who shows the possibility of a suicidal behavior or suicidality would be excluded for safety reasons. And once again, the full list of exclusion criteria are available on clinicaltrials.gov. Next slide, please, Rachel. So I just want to say something about the heart. People living with myotonic dystrophy often have issues with heart function. There are a number of ways, depending on what the issue is, there are a number of ways to address this. It can be with medication. Some people actually have devices implanted to manage heart conditions. When we did the focus group and I had our advisory board that Rachel mentioned, a number of patients that we spoke with told us that they had been excluded in the past because they had an implanted device. And I want to reassure all of you that simply having an implanted device is not exclusionary. Many implanted devices are allowed in the study, particularly if they're designed to prevent heart problems or deal with manageable heart problems. Certain devices that treat very serious heart disease, heart failure, and so on are not allowed. So there are certain heart conditions that are not allowed in the study. Before including you in the study, we do what's called a Holter monitoring on you. This is like a portable electrocardiogram that records your heart rhythm over a 24-hour period while you're living your life away from the office. And we're looking for certain types of abnormalities. But the fact is that many patients with myotonic dystrophy do have abnormal cardiograms, and many of these are permitted in the study. So I want to reassure those of you who might be considering participating in the study that simply having a cardiovascular issue does not automatically exclude you. We are being very careful about who we allow into the study, and we are doing very careful monitoring since this is the first time we are administering this drug to this population, which certainly does have slightly higher cardiovascular issues than the general population. So we're doing this for reasons largely of safety. Next slide, please. So in terms of the study procedure, and I'll go through these relatively quickly, your overall participation in the study from the screening period all the way through lasts approximately 4 months. You'll be asked to have an optional sleep test at the beginning and the end of the study. Again, it's not necessary to do that. As I've mentioned, we'll do a blood test to look for confirmation that you have the genetic signal. We'll ask you to keep a 7-day sleep diary. We'll ask you to do a number of questionnaires related to symptoms and how you're feeling. There'll be standard physical exams, electrocardiograms, blood samples, urine samples, and we will ask you to take the study medication once a day during your morning, and the study investigation, the study investigational material will either be pitolisant at 1 of 2 doses or placebo. Next slide, please. So we have a number of study visits. I won't go into these in great detail, but obviously we have the screening visit. Second visit, we complete any baseline assessments we need and we do the optional sleep test. You'll begin to take your study medication. If you are eligible for the study, you'll be randomized. And we'll ask you to keep a dosing diary where you record when you take your study medication. We will also ask you to bring in your study medication at each subsequent visit. There will be another study visit about 3 weeks later. where you'll have another electrocardiogram and we'll do another Holter monitor. There'll be another site visit with an electrocardiogram and more questionnaires. And at the last on-site visit, you'll have a full physical exam, another cardiogram. If you have opted in, we'll do another sleep test with you. And then if you are eligible and interested, you'll be able to enter the long-term safety study that lasts approximately 1 year. There are also certain telephone visits in between these on-site visits, and there are opportunities should there be a reason that you are unable to make an on-site visit that we will provide assistance for that. We will or we will have certain assessments done in your home. Next slide, please. So these are the sites that have agreed to participate in the study. We certainly understand that many patients who might be interested and eligible to participate are not located in these areas. We have a full concierge service wherein we will provide assistance. We will provide you with transportation, accommodation, meals, and so on and so forth. So, that should not be a consideration. We intend to make this as easy as possible, to provide as much assistance as possible. Certainly should not cost anybody any out-of-pocket money. Everything will be reimbursed if there are out-of-pocket expenses and so on and so forth. You can find a complete list of sites that have been activated on the clinicaltrials.gov website. There will also be an email address which Rachel will provide to you at the end of this presentation shortly, where you can make inquiries, and certainly Harmony will refer you to an appropriate site should you be interested in participating, where you can be assessed and determine whether you're eligible to participate or not. Next slide, please, Rachel. So as I've mentioned, there's an open label long-term extension study. After the double-blind phase is completed, patients would have the option, and it is simply an option, to enter this open label long-term extension phase to continue to help us collect information. And if you meet the criteria for this part of the trial and you choose to continue, you will receive open label, in other words, not blinded, investigational mitigation, and have occasional periodic check-ins with the trial site to collect information, see how you are doing, if there have been any adverse events, and so on and so forth. Next, please. I think that pretty much takes me to the end. I will turn things back to Rachel now, and happy to answer any questions that I can.
Yeah. Thanks so much, Bill. I really appreciate that great overview. And we're going to do a couple of questions. We'll promise we won't keep you all over an hour. And I'm going to take the slides down just to be a little bit more conversational with Bill. But before I do, as Bill promised, if you do have questions about this phase 2 trial in myotonic dystrophy type 1, please reach out to us at clinicaltrials@harmonybiosciences.com. Again, that's clinicaltrials@harmonybiosciences.com, and we hope to hear from you. So, Bill, I stopped sharing. I'll give you a minute to take a breath. I know you shared a lot of wonderful information. There have been some questions that have come in. I think one of the ones that I saw a number of times was, are you changing your enrollment criteria to participate in the trial? And so I think you did allude to this a little bit, but just so everyone is aware, we have made a protocol amendment to participate in the trial, and I'll let Bill share a little bit more about that.
Sure. So there have been a number of small minor administrative changes, but the primary change that we have made, which I think will make it much easier to participate, is we have changed our primary endpoint. Our primary endpoint was originally the objective sleep test, the MWT that I mentioned. And it was proving problematic for certain patients because they simply could not afford the time to do this, and they were otherwise completely eligible. So we have made this an optional thing. We are encouraging our sites and we are encouraging our patients to undergo the test. It's certainly at no cost, and it's not an invasive test or anything, but it is a time-intensive test. So we've recognized that, and as a consequence, we have also removed any specific criteria that you have to meet for that test, although we're still recording the information. So that's the primary change that we've made. We've made a couple of other minor changes, but that should certainly help with recruitment. And frankly, this was based on feedback that we had received from the sites that had been activated and screening patients, as well as from patients who had approached us expressing interest, and once they learned of the time commitment, have said that because of work commitments and so on was something that they couldn't do. So I just want to stress that we are aware of and we are sensitive to feedback, and we're constantly trying to evaluate our studies. Nobody likes to change things midway, but we're trying to find the most efficient studies. We are very much convinced that the DSS will give us the information that we need on the benefit or potential benefit of pitolisant in addressing excessive daytime sleepiness. And of course, we do have a number of other secondary endpoints that will inform us as well.
Thanks, Phil. A couple other treatment questions. So how is the drug delivered, and how often do patients have to take the drug?
Okay, so the drug is a very small tablet. It's what we call a solid oral dosage form, which is pharmaceutical talk for a pill. It's very small. It's taken with some water upon awakening once a day. Unlike some of the other drugs that are available as wake promoting agents, this is a drug that is only taken once a day, taken in the morning, whenever your morning is. And is a drug that is not scheduled by the FDA, which we think is helpful. But again, the administration is relatively simple. And as I say, we do ask our patients to record the administration of the drug to ensure. It's very important for us to ensure compliance. And if we find that a particular patient was on active drug but did not perceive benefit, and we look and we see that the patient skipped 10 days of treatment, there's a possible explanation there.
Thanks, Phil. A couple more. So is pitolisant a stimulant, and is it different from other treatments I might take, such as Provigil or modafinil, for my excessive daytime sleepiness?
Yeah. So pitolisant is not a stimulant. It is a wake-promoting agent, and in that sense, it is similar to modafinil and armodafinil, Provigil and Nuvigil, in that sense only, in that it is wake-promoting, but it is not a stimulant, and as I just mentioned, because of that reason, it is not a scheduled drug. It's not scheduled by the FDA. They have different drugs that go under one of 4 schedules that determine certain things, and that are based on things such as abuse liability. How likely is one to abuse the drug? And in its development, pitolisant has undergone abuse liability testing, and it has been shown to not have potential to be abused. And as a consequence, the FDA made a determination, and this is something they spend a lot of time on, that the drug does not need to be scheduled. So the short answer is no, it is not a stimulant.
Yeah, thanks, Bill. A couple more in the few minutes we have left. How does the drug pass? Does the drug pass the blood-brain barrier? So pitolisant,
yes,
does it pass?
The drug absolutely passes the blood-brain barrier, and its actions, the actions that we are concerned about, are its actions on and in the brain. So it does have to pass the blood-brain barrier, and that's a consequence of its chemical characteristics. There are many drugs that do not cross the blood-brain barrier. Some of the drugs that we're concerned about in this study are antihistamines. There are certain antihistamines that cross the blood-brain barrier and are sedating. They cause you to be sleepy. Those are not allowed in this study. There are other antihistamines that people take for allergy and so on that do not cross the blood-brain barrier, and they are allowed. So yes, pitolisant does cross the blood-brain barrier, and we can actually measure the amount in the central nervous system.
Thanks. And then just a couple trial questions in the 2 minutes we have left. So these are sort of similar. When does the trial start recruiting, and when does the study end? So maybe just a broad answer to that will help folks.
So the trial has been recruiting for, I think we opened our first site about a year ago, but we're still in the midst of opening sites. We anticipate one of the other changes that came about as the change in the primary endpoint is we lowered the number of patients that we need to recruit in this study. We are anticipating that recruitment will be complete by June of 2023. We're hoping it will be completed earlier, but that is our target date. So we're looking at approximately up to 1 year of recruitment, 1 additional year of recruitment. But when this study has randomized a total of 78 patients, recruitment will end at that point.
Gotcha. And then I think the last one is, well, you had answered this one, Bill. It's, do you plan to open participation in the next phase to wheelchair-bound patients or more people with pacemakers? I know you talked a little bit about assisted devices for walking and pacemakers and this being an exploratory study. So if you could just touch on that again.
Sure. So, there's certain things that we cannot answer now. And the answer is going to be dependent on what the data from this study shows us, particularly with the cardiovascular issues. We need to see, is there any adverse impact of pitolisant on patients with cardiovascular issues? We have very stringent criteria for entry and very stringent criteria for monitoring. We will look at the data and make an assessment. And clearly, if there are no safety issues, we will take that into consideration as we design our next study. The same with all of the exclusionary criteria. I mean, there are certain things that we know are not going to change. For instance, we know because of studies that we've done that pitolisant is excreted in breast milk. We have no studies to look at the impact of pitolisant on infants. So I am as positive as I can be that exclusionary criteria will continue into the next study. But again, the simplest answer is we will look at all of our data in aggregate and make decisions on planning our next study based on what we have learned in this exploratory study.
And then the last question, Bill, is on exclusion criteria. So, if someone is using, what's contribute, or what we would think of as an excessive amount of caffeine, and maybe take that down to a moderate, or if they were on a regular sleep medication that you were talking about, and then they maybe washed out or stopped taking that so regularly, would they, what's the best way for someone to figure out, if they fit into, or what changes they could make, if possible, to participate in the study?
So the best way is probably to express interest and be what we call pre-screened, where the site will usually ask a number of questions. Now, many of these things are things that can be adjusted. For instance, if there's a prohibited medication or an excessive amount of caffeine, typically what we like to do is have you discontinue a given medication for 5 what are called half-lives. A half-life is the amount of time it takes for the amount of drug in your body to be decreased by half. So if you have a certain level of drug and the half-life of the drug is 4 hours, in 4 hours after you've taken the drug, you should have half the amount of the peak level in your bloodstream. So we look for most drugs for 5 half-lives of elimination. So if the half-life of a drug you're on is 4 hours, that means 20 hours, so approximately 1 day to wash out. So for some of these drugs, the half-lives are substantial. We typically like to have you washed out, depending on the specific drug, for perhaps a couple of weeks, but there are certain drugs that can be washed out much more quickly. These are issues that are discussed with the investigator, and very often the investigator comes to us and says, I have a patient who is taking whatever the drug is. Are they eligible? How long do they have to be off of the drug? And I will have that discussion with our medical monitor, and we will inform them. So I don't want anybody to think that if they're on a specific drug, they cannot partake in the study. It may require a month of washout or something like that, but we would certainly encourage you to at least make the inquiries to the clinical trial site and learn that way.
Yeah, thanks, Bill. I think that's a good suggestion to have a pre-screen conversation with the site that you may be interested, or maybe close to where you live if you're interested. I think that's all we have time for. I know we're about 1 minute over. So again, just a huge hearty thank you to the Myotonic Dystrophy Foundation for having us today, allowing us to share a little more We're always reachable at clinicaltrials@harmonybiosciences.com. And then we also hope to see some of you at the Myotonic Dystrophy Foundation conference, where we'll have an exhibit table virtually and then also in person. So please come chat with us and learn more. So again, thank you for your time today. Appreciate it.
Thanks very much.
Thank you so much, Rachel and Bill. Thank you for presenting your study so clearly and answering the important questions. EDS has such a significant effect on our community, and we're delighted that you are working to address it right now. Thank you for being very thorough in the presentation. We're thrilled that Harmony is going to join us in September for the conference, and grateful to you for being one of our sponsors. We look forward to hearing more about your trial in September. And with that, I'll say thank you and have a fantastic weekend.
Great. Thank you, Tanya.